Hello everyone, welcome to the third session of Botany Power Hour session, and in today's session we are going to discuss one very important chapter, that is the principle of inheritance and variation. And if you are a NEET aspirant, you know that this chapter counts nearly 24 plus marks in a NEET examination, as there are 6 questions asked from this chapter from previous year. So what we are going to discuss in today's topic: first introduction, then non-Mendelian inheritance, and then very crucial topic, that is chromosomal theory of inheritance. And this is very crucial topic, chromosomal theory of inheritance, because questions are continuously asking on this topic, and this is little bit difficult to understand. Some MCQ also we are going to discuss, PYQ we are also going to discuss in the session. So let's begin with power hour session, principle of inheritance and variation. And in very first slide, I have in front of you 7 character which is given by Mendel, their dominant trait and recessive trait. I am going to discuss the monohybrid cross, test cross and back cross. So first of all, let's discuss about the characteristic. First character which is mentioned here is a stem height. First character is stem height. Dominant character is tall, so represented by the symbol T, and recessive character is dwarf, so represented by the symbol small t. Now another character is a flower position, flower color, pod shape, pod color, seed shape, seed color. So what is exactly that 4, 4, 1, 4, 5, 7, 1? What does code means? Does code represent the chromosome number on which those characters are present? So if we are talking about the stem height, 4 number chromosome. If we are talking about the flower position, on 4 number chromosome this character is present. Flower color on chromosome number 1. Pod shape on chromosome number 4. Pod color on chromosome number 5. Seed shape on chromosome number 7. And seed color on chromosome number 1. And most probably in NEET examination they are going to ask you the question regarding to the seed shape on which chromosome. So how discuss the dominant trait: tall, exile, violet, inflated, green, round, yellow. Those are the dominant character, and recessive character are dwarf, terminal, white, constricted, yellow, wrinkled and green. Always remember one thing: if we are writing the dominant character by capital letter, then the recessive character written by a small letter. Like if we are talking about the flower position, exile flower position A, then terminal is not small t, it is a small a. If we are talking about the flower color, dominant character is violet by V, then a recessive character by small v. If we are talking about any other thing, like a seed color, dominant character we are talking about capital Y, then green, not small green, it is a small y for recessive character. This is the one thing you have to remember about the 7 true breeding varieties selected by the Mendel. And in examination, if they are asking about the how many character Mendel select, 7 character, how many pair of character, 7 pair of character, how many contrasting trait, 7 here, 7 here, 7 dominant, 7 recessive, means 14 contrasting traits, not 7 contrasting traits. If they are asking about character, 7. If they are asking about the traits, 7 dominant, 7 recessive, means how many traits? 14 traits. I hope so you understood this thing. Let's discuss another one slide, that is the question. Very first slide, and in second slide you have a question in front of you. Let's try to solve this question. First statement saying that Mendel studied 7 pair of contrasting trait. Yes, pair means 14, 7 pair is 14, to 7 pair of contrasting trait in bee plant, and proposed a law of inheritance. There are 3 laws of inheritance given by Mendel. First law of dominance. Second law of segregation. Third law of independent assortment. And only one law is universally acceptable, and this law is law of segregation. Law of dominance have some deviation, law of independent assortment also have some deviation. We are going to discuss this topic, but first statement is a true. Statement 1 is correct. Now second statement, 7 character examined by Mendel in his experiment on a pea plant where, look, first they are saying seed shape. Is that seed shape and seed color is present in the chart? Seed shape, yes. Seed color, yes. Seed shape, seed color, 2 character. Okay, fine. So till that statement is correct. Second, flower color, pod shape and color. Flower color, is that pod color is, flower color is present, flower color is present, pod shape present, pod color, okay. Till that point also this statement is correct. Flower position and stem height. Flower position, yes. Flower position and stem height, yes. It is also a correct statement. So statement 1 is also correct, statement 2 is also correct. Let's find out both statement 1 and 2 are incorrect, wrong. Second, statement 1 is correct, but statement 2 is incorrect, false. Statement 1 is incorrect and statement 2 is correct, false. But both statement 1 and 2 are correct. This is exactly our answer. Let's take this statement 1 and statement 2 is correct. So let's discuss the another slide. Coming to the next slide and next question in front of you. Very easy question, very simplified question. They are asking about how many pair of contrasting character in a pea plant or contrasting trait are present and studied by the Mendel in his experiment. 6, 8, 7, 5. How many? Just look at this statement. 1 pair, second pair, third, fourth, fifth, sixth, 7 pair. Any option belonging to our studies? Yes, 7. C is the correct answer. Let's discuss another slide. Another question for you on the same topic, and those are PYQ. Understood this thing? Those are the PYQ. In his classic experiment on a pea plant, means on Mendel's pea plants experiment, Mendel did not use, just remember the thing, the statement lies correct, not correct, incorrect, true, false, not correct, exactly correct. Those are the situations in which student trapped into the examination. So understood the thing they are asking about. Did not. Flower position they use, yes, they used the flower position. They used seed color, yes. Pod length, I remember pod shape is mentioned, pod color is also mentioned, but pod length is not mentioned. So this is not mentioned and they are saying did not use. It means C is the correct answer. Seed shape definitely they mentioned in the experiment. Now directly coming to the topic, that is the monohybrid cross, which is very essential topic of this chapter, and on that basis we are going to study a 2 law. One is law of dominance and second is law of segregation. Let's discuss monohybrid cross. What they are saying in case of monohybrid cross, they are saying a cross involving 2 plants different in one character's pair, like Mendel cross tall and dwarf pea plant to study the inheritance of one gene. Look, if Mendel study a tall and dwarf plant, so here we are taking tall cross with dwarf. Now what is exactly symbols we are using for talls? Not symbols, we are going to say it allele. Okay, so traits are tall and dwarf. Now allele is capital T for tall, small t for dwarf, because they are pure line. Now what type of gamete is formed? So here law of segregation is used. From capital T same, so there is only one gamete capital T, and thus gamete also segregates. So here is a small t, the cross between them, let's cross between them, capital T, small t. What is the characteristic of this plant which is formed? That is known as the F1 generation. Capital T is present here, small t also present here, but it is always appear dominance, because Mendel give us the law of dominance also. So segregation proved by the gamete separation, and law of dominance show by this F1 generation. This is tall. Characteristic of this is tall, but it is hybrid tall. Why it is hybrid? Why it is not pure line? Because it is manufactured. It produced with help of two gamete, which is coming from the pure line tall and pure line dwarf. So this is a tall plant. Now for collecting the ratio, we have to self cross this F1 generation. So my next statement is selfing or self cross of F1 generation. So if we self cross it, capital T small t cross with capital T small t, we have to draw one table. This table is known as Punnett square. This also asked in the NEET examination. The table is known as Punnett square because given by the scientist. Capital T small t, here male one side female, consider anyone as a male, another one as a female. Capital T small t, in first, here I have to complete this table. In first part, here capital T, characteristic is tall. Capital T small t, characteristic is tall. Capital T small t, characteristic is tall. Small t, characteristic is dwarf. Now, understood the thing. There are 3 tall plant and 1 dwarf. When we observe this plant, that is a phenotypically, observation on appearance, phenotypic ratio. Phenotypic ratio. What is the phenotypic ratio? It is tall is to dwarf. How many tall? 1, 2, 3 are tall. 3 is to 1. 1 is dwarf. Phenotypic ratio is 3 is to 1. If we are talking about the genotypic ratio, then capital T is to capital T small t is to small t, because genotypically they are looking like this. They are not observed exactly. Observation is different thing. By looking on someone, we can only identify phenotypic or phenotype, not genotype. So genotypically they are looking like this. So how many capital T? Only 1. So 1 is to how many capital T small t? 2. 2 is to how many small t? 1. So genotypic ratio is 1 is to 2 is to 1. So understood, if we are talking about the monohybrid cross, the phenotypic ratio is 3 is to 1. The genotypic ratio is 1 is to 2 is to 1. If you understood, let's discuss another some cross like a test cross and back cross and test cross, and those are also an important topic. Now first statement I want to write here, a test cross is used to identify unknown genotype. Identify the unknown genotype. Why I am writing this? Because phenotypic ratio or genotypic ratio we are calculating by using test cross are looking like this. If unknown we don't know, but we have to cross this unknown with recessive parents. Now which is the recessive parents? This dwarf is recessive parents. So take the recessive parents here for test cross only. Take the recessive parents as a small t cross with unknown. We don't know about this. Or this small t with unknown. We don't know this is the capital T tall or capital T small t tall, pure line tall or hybrid tall. Just we have to identify this unknown. Now, if after the crossing over or taking the cross on our behalf, if ratio come 4 is to 1, then this unknown is capital T. If ratio coming like 1 is to 1, then this unknown is capital T small t. Let's try to calculate it is true or false. we are taking capital T is to small t. Here only one gamete is going to form. Here only one gamete, and all four are capital T, small t, that is a 4:0 ratio. Now here capital T, small t cross with small t, small t. Here one gamete is capital T, another gamete is small t, another gamete is small t. First cross capital T, small t, tall, small t, dwarf, 1:1 ratio. Understood the thing? Test cross is responsible for identify unknown genotype. For recessive parent, we know that they are having a small height. If they are having a small height, the symbol will be small t. But tall, for tall plant, we are little bit confused. This tall is pure line capital T, or this tall is capital T, small t. For this confusion, means what we can say, for resolve this confusion, we are doing test cross to identify unknown genotype. And back cross is a cross of F1 hybrid with any of parent. Cross of F1 hybrid with any of parent. It means we can write one statement here, very essential statement, hashtag all test cross are back cross, but all back cross are not a test cross. And this is very crucial statement, because they are saying any of parent include dominant parent as well as recessive parent, and in test cross we are crossing it with only recessive parent. So all test cross are back cross, but all back cross are not test cross. So one statement, another statement to identify unknown genotype. Those two statements, this cross is very important as we are talking about the back cross and test cross. I hope so you understood this topic. Let comes to the another some crucial point. Mendel made similar observation for another pair or trade, and he proposed the some factors. Mendel performed his experiment in 18th century, 1856 to 1863. Now, long back, there is no evolution of genetics. There is no evolution of scientific theories. So, he identified gene, but he don't know the term gene. So, he give a term factor as a gene. Now, in today's day, we know the segment of DNA is gene. But at that time, in 1856, Mendel don't know about the gene, so he give the term factor. Factor are now called as a gene. I hope so you understood this topic also. Let's discuss one another very crucial question. This type of question asked into the NEET examination, and 2006, 2007 AIPMT, this question is asked. Now they are saying how many different kind of gamete will be produced by a plant having genotype capital A, small a, capital B, capital C, small c. Let's try to solve this question. You have 30 seconds of time. I am taking water. 30 seconds of time to solve this question. Option is in front of you, 3, 4, 9, 2. I think so you are trying to solve this question. Okay, time is over, 30 seconds is completed. Now, what is exactly a gamete which is going to form? First, we are going to discuss this. Then one short trick to identify this type of question, or identify to answer this type of question. Capital A, small a, capital B, capital C, small c. For the calculation, we are using fork line method. Take a capital A here. Take a capital A here. After that, capital B, both are same. So give us a fork, that is the arrow, capital B. Now after B, capital C, small c are different. means heterozygous, give two arrow, capital C and small c, and produce a gamete. That capital E, capital B, capital C, capital A, capital B, capital C is the first gamete. Capital A, capital B, small c, capital A, capital B, small c is the second gamete. Now, do this same thing with A, small a. Now here, small a, one arrow, capital B, two arrow, capital C and small c, first gamete. Capital small a, capital B, capital C, small a, capital B, capital C, small a, capital B, small c, small a, capital B, small c. Any other possibility for formation of gamete? No. So how many gamete is produced? 1, 2, 3, 4. Any option? 4. 4 is the correct answer. So this is a longest method. Directly, if you want to calculate this answer, you have formula with you, 2 raised to the power n. Number of gamete, what I am saying, number of gamete equal to 2 raised to the power n, where n is equal to number of heterozygous pair, where n is equal to number of heterozygous pair. How many heterozygous pair? 1, 2. So n is equal to 2 in this case. 2 to the power 2 is equal to 4. 4 is the correct answer. By any method you can calculate. I hope so you understood this question. Let's go to the another. The test cross is carried out to assess the number of allele. No, determine whether two species. No, determine the genotype of F2 plant. Yes, determine the genotype. Now, very crucial, very important topic, that is non-Mendelian inheritance. Now, what is exactly non-Mendelian inheritance? What is Mendelian deviation? So, non-Mendelian inheritance or deviation is some point where on the time of Mendel's experiment, which is not studied or which is not taken into the consideration. So those are incomplete dominance which break the law of dominance, codominance which break the law of dominance, multiple allele which breaks the law of Mendel, polygenic inheritance which break the law of independent assortment, Pleiotropy also break the any law, but no, there is no any deviation, no any non-Mendelian inheritance which break the law of segregation. So that's why understood the thing, law of segregation is universally acceptable and law of dominance, law of independent assortment are not universally acceptable because there are some non-Mendelian inheritance or non-Mendelian deviation are present for those two laws. First, incomplete dominance. What is exactly incomplete dominance? Now here it is a inheritance in which the offspring show intermediate character. What Mendel tell us? Tall plant, dwarf plant. First generation always tall. All 4 plants are tall or all the progenies are tall. But they are saying offspring show intermediate character. An example is, just noted down this example. You have to learn this example. Snapdragon, that is a dog flower, Anthracynum species, and Mirabilis jalapa 4O clock plant. If in case of animal, Andalusian fowl are also example of incomplete dominance. Let's cross red and white and they are going to produce a pink. So what exactly Mendel told us? If red, capital R cross with white, small r, there is a gamete formation, capital R, small r, cross, capital R, small r. According to Mendel, this F1 generation is red because dominant character is red and recessive character is white. So according to Mendel, F1 generation is red, but in that case, not theoretically, practically, they are having flower with pink color. So now this is the deviation for Mendel's law of dominance. Here, after selfing of this capital R, small r for F2 generation, we have to self, then capital R small r cross with capital R small r, first cross with cross capital R, first with second capital R small r, second with this capital R small r, last small r. This is red, those are pink, this is white. So a genotypic and phenotypic ratio are same. Genotypic ratio is also 1:2:1 and phenotypic ratio is also 1:2:1. This is happen in case of two things. One is incomplete dominance, second one is the codominance. Codominance is also same. In case of codominance also, this is carried out, this ratio is carried out. Now here in Anthrhynum species, snapdragon, a red flower were crossed with the white. Yes, we just crossed it. F1 generation, pink flower are formed. Yes, when pink flower were self into the F2 generation, show white, red and pink. Yes, this is also true. Choose the incorrect statement from the following. Incorrect statement they are asking. Experiment does not follow the principle of dominance. Pink color in F1 is due to the incomplete dominance. Ratio of F2 generation, it is also true. All three. Law of segregation does not apply in this experiment. Now I told you, law of segregation is universally acceptable. It means in any case, any cross, law of segregation is happen. Now look at the cross. If we are segregating this gamete, it means segregation is applicable. And they are saying in question, law of segregation does not apply in this experiment. This is a false statement and they are asking us the false statement, incorrect statement. So correct answer is D. Codominance, the best example of codominance is a color coat of cow, or we can say that a AB blood group. What is exactly AB blood group? If we are talking about the blood group AB, blood group AB, we take blood from a person which is having blood group AB and observe their RBC in a microscope. So the RBC, for example, I am drawing here one RBC. For example, this is the RBC. On this RBC, antigen A is also present, antigen B is also present, and in the plasma of this person, no antibody. If there is no antibody, it means AB blood group can accept the blood from any of the patient or any of the donor. So because of no antibody, there is no resistance. So that's why AB is universally acceptable. Now understood the thing. They are given us the blood group AB and two antigen A, antigen B is present on the RBC. Both RBC showing this blood group. If antigen A is dominant, then blood group will be A. If antigen B is dominant in this case, blood group will be B. But blood group is not A, blood group is not B, blood group is AB. It means both antigen A and B is dominant. That's why this concept is known as codominance. Now we are not going to discuss so many things, just understood the reason. Blood group A having allele capital IA and I small I. Now blood group A, they are saying having allele IA, or this is a blood group A person, as well as if there is a IA and IO, this IO is equal to small I. In some book they are showing small I, in some book they are showing IO. So let's consider this. This is also blood group A, this is also blood group A, but this is the homozygous and this is the heterozygous. Now for blood group B, IB or IB, IO, blood group AB, only one, IA and IB, both express, and blood group O, both is IO and IO. This is the symbol. We have to remember this symbol. On the basis of that, we are going to solve some MCQ. Let's go to the next slide. They are saying when IA and IB, both allele, are present together, they both express their own type of sugar, and this called as codominance. I told you AB blood group is an example of codominance. Now let's discuss a cross for this topic. From this topic, let's discuss the cross. If we are saying male with blood group AB married with a female with blood group O. What is the question in front of you? Male with blood group AB and female with blood group O. So what is the alleles for blood group AB? IA and IB. Allele for blood group O is IO. Let's try to cross this. If we are saying IB, two type of gamete is going to be formed, IA and IB, cross with only one gamete here, IO. First cross with first, IO. First, second cross with first, IO. And IO means blood group A. IO matlab blood group B. Now the percentage is 50%, 50%. In that cross, the child having blood group A and B with parents blood group AB and O, if there is no matching of blood group with your parents, it means you are not taken from any what we can say from kachre ka dabba. You are not taken from any kachre ka dabba. It is a reason behind that. There is a possibility if parents having different blood groups, child can have a different blood group. Almost time it is matching with any of parents, but if you are not matching, it does not mean you don't have have gene of your parents. Okay, let's discuss the another thing. If in a marriage between male with blood group A and female with blood group B, the progeny had either blood group AB or B, what could be the possible genotype of parent? Very crucial question and important question to understand the topic. In a marriage between male with blood group A cross with a female with blood group B, if we are going by homozygous nature, IA and IB, only one gamete is produced here, IA, and only one gamete cross IA and IB, it means only possibility of blood group AB. So they are saying we want AB as well as B. Now if we are going with A cross with B, A with IA and IO, B with IB and IO, so here possibility of blood group AB, then blood group A also, then blood group B also, and blood group O also. So we need only AB and B. Now if we are going with the possibility A cross with B, IA, IO cross with IB, first cross blood group AB, second cross blood group AB, third cross B, blood group B, and fourth cross blood group B. Only AB and B. We need symbol IO and IB. IB. This is only a correct answer. I hope so you understood this MCQ. If you don't, just go 1 minute back, observe the question, try to solve it. Any of possibility, we need only AB and B. So for this we have to done this process. Next question, very important question. A certain road accident patient with unknown blood group needs immediate blood. Unknown blood group needs immediate blood. His one doctor friend at once offer him a blood. What was the blood group of the donor? A doctor offer him a blood. The blood group of donor. Now we don't know the blood group of patient, and we need to start immediate treatment. At that time we have to use universal donor. So I told you AB is a universal acceptor. Which one is the universal donor according to you? O blood group is a universal donor. So blood group O is the correct answer for that question. Now, third concept, multiple allele. Third concept, multiple allele. It is the presence of more than two allele of, understood the thing. Till that time, we are saying if character is tallness or plant height. What we say, tall or dwarf. If we are saying seed shape, round or wrinkled, and the symbol for that, capital R, small r, capital T, small t. Two allele we are using. Now, if we are saying blood group, just we talk about that. If we are saying blood group, then one allele is IA, one allele is IB, and one allele is IO. A single character controlled by how many allele? More than two allele. A single character is controlled by how many allele? More than two allele. So this is an example of multiple allelism, and ABO blood group is a good example of multiple allelism. Now, on the basis of that, let's solve one more question. ABO blood group in a human are controlled by the gene I, right? It has a three allele, IA, IB, I means IO. Since there are three different alleles, six different genotype are possible. How many phenotype can be occur? They are saying six different genotype. How many? Look, IA is the first genotype. IAIO is the second genotype. Both represent blood group A, means phenotype is blood group A. Now, third IB, fourth IO. Both represent blood group B, means phenotype is B. Third one is IB, represent blood group AB, phenotype is AB. Fourth one IO, blood group is O, represent phenotype blood group O. So how many genotype? Let's calculate genotype. 1, 2, 3, 4, 5, 6, 6 genotype. Yes, they are saying 6 different genotype. How many phenotype? A, B, AB, O. 1, 2, 3, 4. 4 is the correct answer. Now, let's move to the next question. A man with blood group A married with the woman with blood group B. What all possible blood group of their offspring? Let's cross it. Blood group A cross with blood group B. We are going with first homozygous. First we are going with the homozygous nature. IA cross with IB, crossing IA, IB, and this is a blood group AB. Any option with blood group AB? A and B only, O only, AB, O only, AB and AB only. So not any option showing blood group AB. It means we have to go with the another blood group A and blood group B heterozygous nature. Let's try to solve with this. Blood group A cross with blood group B. For blood group A, IA, IO heterozygous, IB, IO heterozygous. First cross with is AB. Blood group A, blood group B, blood group O, all possibilities. Any option with blood group A, B, AB, O, this second number option B is the correct answer. Let's go to the next question. The genotype of a husband and wife are IA, IB, IA, IA, IB, blood group AB, cross with IA, IO, blood group is A. Among the blood type of their children, how many different genotypes and phenotype possible? Let's try. First cross with first IA or IA, that is blood group A. Second cross with IA, IO, blood group A. Third IA, IB, blood group AB. Fourth IB, IO, blood group B. So all 1, 2, 3, 4, 4 genotype. 4 genotype. Those are genotype. All 4 are different. But phenotype, those are same. 1, 2, 3. 3 phenotype. 3 phenotype, 4 genotype. Any option? 3 genotype, 3 phenotype, 4 genotype. 4 genotype, 3 phenotype. Yes. Option number B, 4 genotype and 3 phenotype. Let's come to the next topic, that is a polygenic inheritance. What is exactly polygenic inheritance? Polygenic inheritance in which some traits are controlled by several genes. Means, look, there is a one character controlled by many genes. And well known example of this is human skin color. We are living in India. We are having skin color, intermediate skin color. We are not black. We are not white. We are not very fair. We are not very dark. We are having intermediate skin color. And intermediate skin color is represented by, look, assure 3 genes, capital A, capital B, capital C, control the human skin color. Only one character, how many genes? 3. Dominant form, capital A, capital B, capital C, represent the darker color. Means capital A, capital B, capital C. What is the alleles for this? Capital A, capital A, capital B, capital B, capital C, capital C. Showing the dark color. Small a, small a, small b, small b, small c, showing the light color. Now I'm saying our color is intermediate. Not very dark, not very light. Not very fair, not black. So for intermediate, it is capital A, small a, capital B, small b, capital C, small c. I have one question for you. If I am saying capital A, small a, capital B, small b, capital C, small c, those are the genes. Find out how many number. Find out number of gamete. Find out number of gamete. I am not asking about the type of gamete. You don't have to do the 4th line method and find out the type of gamete. Number of gamete I am asking. 30 seconds only. Look, understood the thing. If we are saying heterozygous pair, how many heterozygous pair? 1, 2, 3. 3 heterozygous pair. N is equal to 3. Answer is 2 to the power n. 2 to the power 3 is equal to 8. How many number of gamete? 8 number of gamete. Let's move. This is all about the polygenic inheritance. Pleiotropy, just reverse. A single gene control many character. Very known example is star synthesis in P, phenylketonuria and sickle cell anemia. I am going to just little discuss about the sickle cell anemia. What is happening in case of sickle cell anemia? There is alteration in the beta chain of the RBC, beta chain. And due to this alteration, due to these changes, due to this mutation, which is known as the point mutation, two character change. Which are the two character? One is change in shape of RBC. Second, change in beta chain structure. If one gene, pleiotropic gene, is responsible for changing two character, then this is known as the pleiotropy. Now, some students are very confused in this topic. They are assuming one gene control many protein. That's why this is happen or this is takes place. Phenylketonuria, sickle cell anemia. No, this is false. One gene produce one protein, and this one protein is produce one enzyme, and this enzyme have multiple role, and due to this multiple role, pleiotropy is takes place. So this gene which is produce one protein, one enzyme, this gene is known as pleiotropic gene. So we don't need this slide now. Let's delete this and go forward. Question for you: dominance, codominance, pleiotropy, polygenic, and the column. Let's discuss. Dominance. Many gene govern a single character. No, many gene govern a single character is the polygenic inheritance. In heterozygous organism, only one allele express is spread. Okay, it is, we can say, dominance. In a heterozygous organism, both allele express, codominance. Single gene influence many character is pleiotropy. So A2, B3, C4 and D1. 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1. B is correct answer. Which of those are represent the inheritance of blood? Obviously, multiple allele codominance and as well as dominance ABC. ABC, 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 ABC is the correct answer. The best example of pleiotropy: star synthesis in P, phenylketonuria, sickle cell anemia. Any option? Phenylketonuria. In cross between male and female, both heterozygous for skin color. Okay, a cross between male and female, both heterozygous for sickle cell anemia, means HbA, HbS cross with HbA, HbS. Both are carrier, right? Both heterozygous for sickle cell anemia. What percentage of progeny will be diseased? This is normal, this is carrier, this is carrier, this one is affected, 25%. Any options? C is correct, because those are 50, 25, so 25 affected. We are coming to the next topic. Why did Mendel work remain unrecognized? Look, in today's date, we have any news? If we have any news, we are going to call our best friend. Oh, this has happened exactly. Or otherwise we send a reel on Instagram, or we have WhatsApp, we have various sources. So that's why communication is easy in today's date. But when Mendel performed his experiment like in 1856 to 1863, communication was not easy. Communication is not that easy. Mathematical approach. Now you are a biology student, right? And any student from JEE came to you and he told you that this is exactly a phenomenon related to the biology. You told him, hey buddy, next time, better luck next time. I am a biology student and you are JEE aspirant. Like the same way, on that time, Mendel used mathematical approach to solve a genetics. That's why scientists are not happy with that experiment, but experiment is so good. Experiment is almost up to the mark. On that time, before 200 to 250 years, this is the best experiment. But still, scientists didn't accept this experiment. Concept of gene, factor, stable or discrete unit is not explained by the Mendel. He could not give physical proof of this existence, so that's why the Mendel is remain unrecognized, but he told one sentence in his language that Meina Zeit Viard Sean Komain. That means my time will come. He say apna time aega, in short. Okay, so let's go to the another. Which one of those given below are the period or Mendel's hybrid 1856 to 1863? Yes, this is the correct answer. Now, so after 30-40 year, on 90s, Hugo de Varies, Coren, Tesmark, independently on their particular countries, in their particular countries, rediscover Mendel's result, and they concluded that Mendel was so good in his studies. That's why after those 90s, Mendel is awarded for Nobel Prize also, but he's not alive at that time. So what he is going to do with this Nobel Prize? But today's date, in live session, in the power hour session, we are discussing about the Mendel. That's why Mendel is known as father of genetics. Mendel is known as father of genetics. Who is the father of experimental genetics? T.H. Morgan is the father of experimental genetics. Now, chromosomal theory of inheritance given by Walter Sutton and Boveri on 1902, and this is proven by Morgan. So they are asking about the given proposed by Sutton and Boveri, and if they are asking about who is what we can say giving us the experimental proof or prove the chromosomal theory of inheritance, then T.H. Morgan is correct answer. Given by Sutton and Boveri, proved by T.H. Morgan. Now, question: Mendel law of independent assortment does not hold good for the genes that are closely located on the same chromosome. True. Closely located genes are assorted independently. Not possible at all if there is chromosome and gene A and gene B is present very close to each other. How can they assort independently? Not possible at all. So one assertion is true, reason is false, assertion is true, reason is false, assertion is true, reason is false. Now, T.H. Morgan performed the experiment on Drosophila. Why he select Drosophila? Experimental verification was given by Morgan. Why he select Drosophila? Simple synthetic medium for growth, short generation time of Drosophila, breeding is easy, 100 of progeny at the same time, easily distinguish, easily identify male is smaller, female is large, and hereditary variation are shown in the microscope. Now, what exactly he studied? One is the linkage, second one is recombination. In short, third one is also independent assortment. Look, if we are saying independent assortment, then all genes are present on different chromosome, capital A, small a, okay, and capital B, small b. It means we can go by capital A, capital A, capital B, capital B. Capital A, small a, capital B, small b. Capital A, small a, small a, small a, small a, small b, small b. Those are parental type, those are the recombinant type. If we are saying about the linkage, linkage means they are closely located. Capital A, small a, capital B, small b, closely located. So, independent assortment, in short, I am going to explain you, independent assortment having possibility of 50% recombination, highest. Crossing over having ability to 0 to 50% recombination, and linkage is very less. In linkage, very less recombination takes place. In experiment, we are going to discuss this. Those are the tightly linked gene and those are the loosely linked. So crossing over takes place in this gene. The possibility of crossing over highest in this link because they are far away from each other. By Morgan's experiment, we are going to study this 3 character on X chromosome. This is the X chromosome. Body color, eye color, wing size. If we cross those 2 characters which are close, there is more linkage, less recombination. If we cross those two characters, that is the eye color and wing size, what we are going to conclude it? More recombination, less linkage. Same, body color, eye color. If we are talking about this experiment, and this one is the body color and eye color, gene are located very close to each other. That's why parental type is 98.7% and recombinant type only 1.3%. But if the gene are located far away from each other, like eye color and wing size, which is away from each other, which is having distance, so parental type is 62.8% and recombinant type is 37/2%. I told you recombination can't be exceed more than 50. If only independent assortment, there is a 50% recombination. Other than that, if there are genes present on the same chromosome, if they are far away also, maximum recombination can be takes place 50%, not more than that. So the cross B represent recombination and cross 1 represent a linkage. Cross 1 represent linkage. Recombination if takes place, it need crossing over, and my simple question is to you, in which stage of meiosis 1, prophase 1 crossing over takes place? It is Pachytane or Pachytane. Now, the term linkage was coined by T.H. Morgan. Those are the conclusion of linkage. Those are also conclusion. Now, this is also an important topic. Alfred Sturtevant, he is a student of Morgan. Alfred Sturtevant is a student of Morgan. Use the recombination frequency between gene pair for measuring the distance between the gene and mapped their position. He give us 1% recombination is equal to 1 centimorgan unit. So if there is 70% recombination, there is a 70% centimorgan, 76 centimorgan distance. 8% means 8, 9% means 9. So those two genes are close to each other. It means in between them there is a linkage. Those are little bit far, so in between them is recombination. And in between, if we are talking about, Mendel didn't perform the cross between wing size and body color, but if he take those two genes, maximum recombination is shown because those are far away from each other. This is a simple logic, and he is a Alfred Sturtevant. Genetic map are used as the starting point in the sequencing of genome, that is the human genome project. In next topic, I am going to discuss with you about the human genome project also, because we are taking molecular basis of inheritance on which day? On 1st of April. Genetics, I am taking one chapter, one day, because genetic is very vast. 6 by 6 means 6 into 6, 6, 6, 12 question. 12 question count 48 marks. 48 marks is very crucial for NEET. So for revision purpose, for this 1 hour experiment, I am going to take genetics is little slowly. After that, I am going to cover a chapter little bit faster. Now, the question, the frequency of recombination between gene pair on the same chromosome as a measure of the distance between the gene was explained by T.H. Morgan? No, he is the father of experimental genetics. Gregor Mendel? No. Alfred Sturtevant? Yes, Alfred Sturtevant is a student of Morgan, and he give us the same frequency of recombination. Sutton Boveri? No. Sutton Boveri is, who is Sutton Boveri? He give us the he proposed the chromosomal theory of inheritance. So C is a correct answer. Last question, the map unit is adopted in the construction of the gene. 1 unit distance between two expressive genes represent 100%. No, 1%. 1 unit is 1%. This is the correct answer. I hope so you understood today's session also. This is one short revision of principle of inheritance and variation by Mukul sir, botany faculty, PW Vidyapeeth. Thank you so much for joining. Let's meet in the another session. Bye bye and take care, guys.